Dear students, as you know, the structure of the RNA is formed as a result of the folding of the primary structure onto itself. These secondary structures, they lead to the formation of tertiary structures, and some of them, they are pseudoknots. We would like to see how to detect these pseudoknots by using a graphical representation. These pseudoknots can be detected by a simple diagram called the circular graph. This can help us to detect the pseudoknots in a very simple and straightforward manner. Let us see how we can construct a circular plot by looking at an RNA tertiary structure. So given an RNA secondary structure like that, which comprises of the hairpin loops, interior loops, the junctions or the intersections, and bulges, how can we find out that pseudoknots exist within this structure? So the straightforward way is to simply label each nucleotide with an index as shown here by B1, B2, B3, B4, B5, B6, and B7, and so on and so forth. So once you label all of these nucleotides, starting from the first one till the last one, which in this case is represented by Bn, then you can proceed to draw a circle like this, and then you can place each nucleotide starting from the first one on its circumference parameter. Let's see. Let's draw the ticks representing each nucleotide in this RNA structure like that. Now you can start by labeling it by B1, 2, 3, 4, 5, 6, and so on, and towards the 3' end, which is this side, versus the 5' end, which is on the other side, you can arrive at Bn, n-1, n-2, and so on and so forth. So once you have drawn a circle and labeled all the nucleotide bases that are represented by ticks on the circumference of this, then you can start coupling by drawing arcs from the nucleotide bases which are coupled to another nucleotide base. In this case, B1 is coupled with Bn-1. So an arc needs to be drawn from Bn till Bn-1, like that. As you can see, Bn does not have an arc because it is not coupled to anybody. And then you can continue on and you can draw arc between B2 and Bn-2, like that. And B3 and Bn-3, like that, and so on and so forth. So once you have drawn arcs between the coupled nucleotides present in the RNA secondary structure, then you will have something like this in front of you. The important thing here is that all of these arcs, they start from a nucleotide and they end at the complementary nucleotide. However, if one of the arc intersects with another arc which is going from some other nucleotide to its complementary nucleotide, then this represents a pseudoknot. Let us see this here. So as I just made the diagram for you, B1 has an arc to Bn-1, B2 has an arc to Bn-2, B3 has an arc to Bn-3. However, if B7 were to have an arc from B7 to Bn minus 4, and B6 were to have an arc that was intersecting the arc going from B7 till Bn minus 4, then this intersection would represent a pseudo knot. So dear students, if you construct the circular diagram for an RNA secondary structure, and if you see two arcs that are intersecting each other, then you can call it as a pseudo knot.